Okay, students, think about this for a moment. Our body is constantly surrounded by microorganisms. They are in the air, water, food, and on the surfaces we touch. But does that mean that every microorganism causes disease? No. In fact, many microorganisms live naturally on and inside the human body without causing disease. Some are even beneficial to us. Today, we are going to explore the fascinating relationship between microorganisms and human diseases. Hari? Microorganisms and diseases. Microorganisms and human diseases. Normal microbiota. Normal microbiota of the human body. Generally, humans are considered free of microorganisms. Generally, humans are considered free of microorganisms at birth. However, during birth, the newborn first comes into contact with microorganisms associated with the mother. These commonly include lactobacilli. Lactobacilli begin to colonize the intestine of the newborn. After birth, many other microbial populations gradually become established on the surface and inside certain regions of the body. These microorganisms are collectively called normal microbiota. So, normal microbiota are microbial populations that normally live on or within the human body. Normal microbiota are the microorganisms that naturally live on or inside a healthy human body without causing disease. It is very important. Normal microbiota does not cause disease. However, the internal tissues of a healthy human body are normally free of microorganisms. Where are normal microbiota found? Skin. Skin. Then mucous membranes of the nose, thorax, upper respiratory tract, intestinal tract, and genitourinary tract. So, students, remember this important point. Microorganisms can normally live on body surfaces and certain internal surfaces, but healthy internal tissues are normally microorganism free. Humans are generally free of microorganisms and at birth, during normal vaginal birth, the newborn first comes into contact with lactobacilli present in the mother's vagina. These bacteria colonize the intestine of the newborn. After birth, many other microorganisms establish on or inside the body. Common sites of normal microbiota. Skin, nose, thorax, upper respiratory tract, intestinal tract, genitourinary tract. Internal tissues of a healthy human body are normally sterile. Sterile means free of microorganisms. No microorganism. So what about the importance of normal microbiota? The human body contains an enormous microbial population. According to the data, a human body contains approximately number of human body cells 1 to 10 to the power 13. Microbial cells 1 into 10 to the power 14. These are about 10 times more microbial cells than human cells. No, the majority of these microorganisms are harmless. Majority are harmless or beneficial. Example, E. coli, Escherichia coli. Small population of E. coli in the large intestine can help prevent colonization by pathogenic bacteria such as Salmonella typhi. Then synthesize vitamin K, synthesize some B vitamins. These vitamins can be absorbed into the bloodstream and used by body cells. Can you understand? Human cells versus microbial cells. Human body approximately contains 10 to the power 13 human cells, and microbial cells 1 into 10 to the power 14. There are about 10 times more microbial cells than human cells. Importance of normal microbiota. What are the beneficial roles? Prevent colonization by pathogens. Example, Escherichia coli. E. coli in the large intestine prevents colonization by Salmonella typhi. Second importance, produce vitamins. E. coli synthesize vitamin K and some B vitamins. These vitamins are absorbed into the bloodstream. So next, another important concept is probiotics. Probiotics. Actually, probiotics are live microbial cultures. They are live microbial cultures that exert beneficial effects. Example, yogurt. They help certain lactic acid bacteria represents the probiotic. Probiotics are live microbial cultures that provide health benefits when consumed. Example, yogurt. Yogurt contains lactic acid bacteria. What are the benefits of lactic acid bacteria? They reduce diarrhea. They reduce diarrhea. They prevent colonization by Salmonella enterica. Usually, probiotics prevent colonization of Salmonella enterica. They help restore normal intestinal microbiota after antibiotic therapy. So microorganisms are not always our enemies. I think some microorganisms can actually contribute to maintaining health. Next, opportunistic pathogens. Opportunistic pathogens. Although the majority of normal microbiota are harmless, under certain conditions, some may change their interaction with the human body and cause infection. These microorganisms are called opportunistic pathogens. Microorganisms that are normally harmless but cause disease under certain conditions. Normally harmless, but under certain conditions, they cause disease. Example, E. coli, Escherichia coli. E. coli is generally harmless while it remains in the large intestine. When it remains in the large intestine, no any effect. They are actually harmless. However, if it enters other parts of the body, it may cause disease. Example, where large intestine normally harmless, but when it enters to the urinary bladder, it causes urinary tract infection. If it enters to lungs, it may can occur the pulmonary infection. The key idea is a microorganism that is harmless in its normal location may become pathogenic when it enters other body site. Next, terms related to infectious diseases. First, pathogen, the term pathogen. Pathogen is an organism or entity. It is an organism or non-living infectious entity. What is the special ability? Capable of causing diseases. An organism or non-living infectious entity of capable of causing disease. This includes non-cellular entities such as viruses and prions. Next, host. An organism or yes, actually it is an organism. Host is an organism. The organism in or on. In or on. Within or on. Within or on. In or on. Which pathogen lives and multiplies. Host is the organism in or on which pathogen lives and multiplies. Next, pathogenicity. Pathogenicity. Pathogenicity is the ability of a pathogen to cause disease in a host by overcoming the host's defense mechanisms. By overcoming the host's defense mechanism, the ability of a pathogen to cause disease. Parasite is an organism or entity that lives on or in another living organism. An organism or infectious entity living on or inside other organism, which is the host, and obtains nutrients and other resources from it. And these points are very important. They are structured the same. Example, define the term pathogen, define the term host. Write the definitions: pathogen, host, pathogenicity, and parasite. Okay, next. Characteristics of pathogenic microorganisms. Characteristics of pathogenic microorganisms. Pathogenic microorganisms may possess several important characteristics. First one, growth requirements. Suitable or optimum growth conditions. Suitable growth requirements. The optimal growth conditions such as temperature may correspond to conditions within the host body. Next, structure for attachment and protection. Example, capsule and villi. Those are the important characteristics which can be seen in pathogenic microorganisms. Next one. Okay. These are special characteristics of pathogenic microorganisms. First characteristic is what? Optimum growth conditions similar to host body. Second characteristic, presence of structures for attachment to the host cells. What are the examples for structures? Capsule and villi. Third one, ability to produce toxins. Pathogens may produce endotoxins and exotoxins. Fourth one, production of enzymes that promote invasiveness. Hari? Enzymes that help invasion means production of enzymes that promote invasiveness. Examples for such enzymes: phospholipase, lecithinase, and hyaluronidase. Phospholipase, lecithinase, and hyaluronidase. Fifth one, production of other enzymes. For example, DNase. DNase, such enzymes may help alter host processes. Those are the five characteristics of a pathogen, a pathogenic microorganism. Okay, next one, virulence and virulence factors. Virulence and virulence factors. So, what do you mean by the virulence? Now, students, we need to distinguish between pathogenicity and virulence. Pathogenicity and virulence. So, virulence means it is the degree of pathogenicity of a pathogen. Degree of pathogenicity of a pathogen. Some pathogens are highly virulent, while others may be less virulent or non-virulent. Highly virulent chickenpox virus, less virulent mild pathogens. What are the virulent factors? Certain genes of pathogenic microorganisms encode factors that provide the ability to infect the host and cause disease. These are called virulence factors. Virulence factors can help pathogens invade host tissues, colonize the host, overcome or escape host defense mechanisms, and cause the disease, which means disturb normal body functions. The relationship between the host and pathogen is dynamic. Therefore, the outcome of infection depends mainly on virulence of a pathogen and effectiveness of the host defense mechanisms. Okay, there are two major mechanisms of pathogenicity. Two major mechanisms of pathogenicity. First one, invasiveness. First mechanism is what? Invasiveness. So, invasiveness is the ability of a pathogen to invade tissues, by overcoming host defense mechanisms. Invade tissues, then multiply and establish colonization. So, for this, several extracellular enzymes contribute to the invasiveness. Example, phospholipase. Phospholipase destroys animal cell membranes. Next, lecithinase. Lecithinase hydrolyzes the lecithin component of lipids in cell membranes. Third one, hyaluronidase. Hyaluronidase breaks down hyaluronic acid, cementing substance. Hyaluronic acid is a cementing substance between cells, thereby facilitating the spread of pathogens through tissues. Phospholipase destroy animal cell membranes. Lecithinase hydrolyzes lecithin in cell membrane. Hyaluronidase breaks down hyaluronic acid, destroys the cementing substance between cells, helps spread through tissues. Next one, what are the entry portals of entry? Pathogenic microorganisms may enter through wounds of the skin, skin wounds, wounds of the skin, next respiratory tract, next gastrointestinal tract, and genitourinary tract. Okay, the second one, second major mechanism of pathogenicity is what? Toxigenicity. Toxigenicity. So, toxigenicity is the ability of microorganisms to produce biochemical substances called toxins. Toxigenicity, Genicity means what? Toxigenicity means ability to produce toxins. Right? Ability of microorganisms to produce biochemical substances called toxins, which disturb the normal functions of cells. Toxins may be proteins or lipopolysaccharides. They produce specific harmful effects on the host and can therefore be considered biological poisons. What are the characteristics of endotoxins? They are lipopolysaccharides. You have to differentiate endotoxins and exotoxins later. Therefore, now learn about endotoxins, characteristics of endotoxins. First one, endotoxins are actually lipopolysaccharides, and endotoxins are part of gram-negative bacterial cell walls, and they're released when bacteria die. They are heat stable, heat sensitive or thermostable. Can you understand? They are lipopolysaccharides. They are part of the microbial cell. Microbial cell means the bacterial cell wall, and they are thermostable and released mainly when bacterial cells die and cell wall breaks apart. And very important, they are produced only by gram-negative bacteria. Possible effects include fever, chills, weakness, generalized ash shock, serve causes. Shock or death-like serve causes. Example for endotoxin, a lipopolysaccharide of Salmonella type. Salmonella type. So that's all about endotoxins. Now exotoxins. Exotoxins. Can you remember the characteristics of endotoxins? If you can remember, they are lipopolysaccharides, but exotoxins are proteins. Exotoxins are proteins, and they are secreted by living bacteria or released after cell lysis, and mostly they are enzymes, very potent and heat pliable and heat sensitive. They are produced inside bacterial cells during growth and metabolism. They are secreted or released into the surrounding environment. Proteins, they are proteins, they are often enzymes. They are highly harmful even in small amounts because of their catalytic nature. They are proteins. No? So generally, they are thermostable and may be inactivated by boiling. So exotoxins mainly produced by gram-positive bacteria and a few gram-negative bacteria. Types of exotoxins are neurotoxins, enterotoxins, and cytotoxins. First, neurotoxins, they interfere with the normal transmission of nerve impulses. Example, toxin produced by Clostridium tetani. Next one, enterotoxin. Abnormally stimulate cells of the gastrointestinal tract. Example, toxins produced by Vibrio cholerae. Third one, cytotoxin. Damage or kill host cells through biochemical of enzymatic effects. For example, toxins produced by Corynebacterium diphtheriae.